Hello, my name is Samuel Billig, and this is my presentation titled A Time Course of Immunomodulatory Effects of Stress in the Northern Leopard Frog Tadpole. So in this research, we used the species Lithobates pipiens, and we were assessing the innate and adaptive immune responses of these tadpoles. We did this using the bacteria pathogen Aeromonas hydrophila, and we stressed these tadpoles using the stress hormone corticosterone. We chose the northern leopard frog for a couple of reasons. Lithobates pipiens is found throughout the Midwest and can be found in wetlands throughout Illinois. Additionally, the species is frequently used in laboratory research and was used in the research we actually built this project off of. So Aeromonas hydrophila was the bacterial pathogen we chose. It's a gram-negative bacteria, and it's a common opportunistic pathogen in amphibians and fish. We chose this species for a couple of reasons. One of the big ones being it's easy to identify, and it has some very visually recognizable features. One being red leg syndrome, which is a red discoloration of the legs and abdomen of afflicted organisms. Additionally, this is something that this species would naturally be exposed to in wild populations. We use corticosterone in simulating a stress event for our tadpoles. Corticosterone is the primary stress hormone in many lower vertebrates, including amphibians like the northern leopard frog tadpole. And corticosterone is a glucocorticoid, meaning its release has a direct effect on energy stores in the body. When glucocorticoids are released into the body, it frees up energy for systems that need that energy in the moment. Innate immunity is our body's initial protection for anything that might be trying to harm us, including pathogens or parasites. It's composed of physical barriers like the skin or mucous membranes, as well as smaller components like the complement cascade system and antimicrobial peptides. All of these things together help your body prevent invasion by foreign parasites or pathogens. And previous research has indicated that the quality of the food source for the organism can be a major factor in the innate immune integrity. So the two primary components of the adaptive immune system are the cell-mediated parts and the humoral parts, which concern antibodies produced by B cells. We focused on the humoral components in our research, and we were building off prior work done by Weber et al. In 2017, in which they found that the adaptive immune system of these tadpoles was impacted by the presence of a corticosterone stressor. We had a two-part hypothesis for this research, the first being that the innate immune response would be reduced in the experimental group after an extended exposure to our corticosterone stressor. And our second hypothesis was that the adaptive immune response would be reduced over time when exposed to a chronic stressor. Our research began in the fall of 2018 with the arrival of 96 tadpoles from Carolina Biological Supply Company. We separated our tadpoles into two groups, an experimental and a control group, housing two tadpoles in a container of 500 milliliters of water, and we had 22 containers for each group. We performed partial water changes for the first week to allow the tadpoles to get used to their environment, and those occurred every 2 days. Then for the remainder of the study, we did water changes every 3 days. Water temperature was about 22 degrees Celsius, and it was aged prior to water changes occurring. The tadpoles were fed a high protein food called Zoomed after each water change, and they were also dosed. The experimental group received corticosterone dissolved in ethanol, as we used ethanol as a medium by which we delivered the actual corticosterone to the tadpoles, and the control group received an ethanol control. We began sampling our tadpoles at 8 weeks. We placed them in an anesthetic bath containing MS-222 or Tricaine S, which is a commonly used fish paralyzer. We took samples from the tadpoles by making an incision near the base of their tail, severing their caudal vein, and collected blood using capillary tubes. We then placed the capillary tubes on a centrifuge and centrifuged the samples to isolate the plasma containing the innate immune components that we wanted to actually analyze. After collecting our samples, we performed a bacteria killing assay using the plasma from the samples and the bacteria Aeromonas hydrophila. In the spring of 2019, we began the second part of our study with the arrival of 70 tadpoles from Carolina Biological Supply Company. All of the tadpoles in this part of the study were dosed with 100 microliters of the bacteria A. hydrophila, and the control group was dosed with 125 microliters of ethanol, while the experimental group was dosed with 125 microliters of a corticosterone ethanol mixture. So we began sampling these tadpoles immediately on their arrival. We sampled 10 of the individuals from the group to determine a baseline value for the antibody presence of these tadpoles and their exposure to previous pathogens. For the remaining 6 weeks of the study, we sampled 5 individuals from each group. In our analysis of our samples that we collected during the second part of our study, we performed an ELISA using a rat serum and lyophilized A hydrophila. We used secondary antibodies to identify the presence of IgM and IgY from our tadpole populations, and we analyzed our samples at 450 nanometers. So in analysis of the results from our assay from the fall of 2018, we found that there was a slight increase in the pathogen killing ability of our experimental group of tadpoles. So those tadpoles that were dosed with the corticosterone or the artificial stressor had a slightly increased ability to fight the presence of a pathogen compared to those that had no exposure to an additional stressor. In analysis of the data we collected in our ELISA, we found a couple of things. The first being that the IgM presence in our corticosterone group was a little bit lower than that of our control group. However, it was not statistically significant. So if you look here at 14 days to 21 days, you see that our corticosterone dose group, the black line there, is a little bit lower for that IgM production. However, our significant finding was that the production of our IgY, or that a secondary targeted antibody towards the pathogen presence, was significantly reduced in our corticosterone dose group compared to that of our control group. And if we go ahead and look here at this red line compared to the green, we see not only was the production of that IgY reduced, its peak at 35 days being lower than the peak there at 28 days for the control group, but also it took almost a week longer for the tadpoles that have been stressed or artificially stressed with the corticosterone to produce that secondary response effectively. So we know stress affects innate immunity from previous research, and we know that stress's influence on innate immunity can also be affected by the food quality that the tadpoles receive. Specifically, tadpoles that receive a high quality, high protein food are able to better endure stressor and mount a more effective immune response. So we think that what might have been going on with the first part of our study, where we saw the stressed tadpoles outperform the bacteria killing ability of the non-stressed tadpoles, was that those tadpoles were able to eat enough food to compensate for the demands of their corticosterone presence, and thereby their systems had more energy available as it was being freed up by the corticosterone, but they were also able to make up for the amount of energy they were expending by eating more food. The second part of our hypothesis was supported, as we found that corticosterone did not appear to have a major effect on the IgM production in our tadpoles. It did seem to be a little bit less intense at its peak, but it wasn't statistically significant. However, we did find that corticosterone had a major impact on not only the production and peak of the IgY response, but the point at which that response began to develop. So we saw a delayed response in producing IgY, and it never reached the same peak as the control group's IgY. So overall, we found that Lithobates pipiens' innate immunity was not negatively affected by the exposure to a corticosterone stressor. Now, we're not sure that this is the end of the story. We think that maybe if Lithobates pipiens is continually stressed over an extended period of time, that innate immunity might be broken down by the presence of that stressor, especially in the absence of a good food source. This is something that we just need to do farther research on and more investigation. We did find that corticosterone had a negligible impact on the production of IgM. However, there was a significant impact on the IgY response of our corticosterone-dosed tadpoles. Not only was the amount of IgY ultimately produced by these tadpoles affected, but it was also delayed in its production in comparison to the control group. So this shows us that stress does have an effect on the adaptive immune system of these individuals and may cause the secondary antibody production to be suppressed when they are chronically stressed. So we know that stress has a differential effect on the innate immunity of Lithobates pipiens, and we've demonstrated that it has a considerable effect on both the development and the potency of the adaptive immunity in this species. So by doing the presentation this way, it's actually kind of hard to take questions on the spot, but if you do have any questions, feel free to send me an email at sbillig@millikan.edu, and I'd be happy to answer or discuss this presentation with you. Thanks for taking the time to listen to my research, and I just give a shout out to Dr. T for all his help during this research project and in the lab and over my past couple years here at Millikan. Really appreciate it.